Hello, everyone, and welcome to today's webinar, an introduction to liquid chromatography-mass spectrometry for the clinical laboratory. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and is made possible by an educational grant from Agilent. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Adam J. McShane from Cleveland Clinic. Dr. McShane obtained his PhD in biological chemistry at the University of Connecticut and completed his clinical biochemistry fellowship at Cleveland Clinic. He currently serves at Cleveland Clinic as the medical director of the Automated Chemistry Laboratory and Toxicology and Proteomic Specialist in the Chemistry Laboratory and as the associate director of the Clinical Biochemistry Fellowship Program. Dr. McShane is a diplomate of the American Board of Clinical Chemistry and an associate fellow of the AACC Academy. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. McShane, you may now begin your presentation.
Well, thank you very much for the introduction, and thank you for inviting me today to speak on probably one of my favorite subjects to speak about, liquid chromatography and mass spectrometry. And thank you to the audience for joining us today, and let me wish you the happiest of April Fool's Days, I guess. So today, we're going to touch on 4 different portions. We're going to talk about the fundamentals of liquid chromatography for the beginner, then move on to the fundamentals of mass spectrometry, some examples of why LC mass spec is important for the clinical laboratory, and then finally, we're going to have an example of a LC mass spec clinical validation. So some of you may be familiar with chromatography from back in your undergraduate or graduate days. I remember we performed forensic type of experiments where we'd have different pens, and then the pen of the person who wrote the ransom note we're trying to decipher. In this experiment, we were putting a dab or writing on the piece of paper with the 3 different pens, and then placing them into a solvent system. The solvent would wick up the paper, and then you would see the black pens would then break down or fractionate into their constituents of the colors that made the black ink. And then you could be able to pinpoint what pen was used by the kidnapper, for example. Another example, as seen here, is column chromatography, often used in organic chemistry. Here, we have depicted in A glass column filled with silica. Silica here is representing a stationary phase. Much like the paper for the pen experiment, this phase does not move. Once the analyte of interest is loaded, here is seen as the purple color, a mobile phase is introduced. For liquid chromatography, the mobile phase is liquid, of course. If you change the mobile phase to gas as the carrier, then you're doing gas chromatography. The mobile phase is introduced at the top of the column. You have a differential partitioning between the mobile phase and the stationary phase, hopefully, to reveal that the purple ingredient is made of blue and red, as it's seen to develop in B and C. Finally, you achieve separation in D, collecting the red portion in E and the blue portion in F. In the clinical laboratory, we don't perform column chromatography. Maybe some labs used to perform thin layer chromatography, similar to the pen and paper experiment, but we're helping with machines to get the separation we need. So we have liquid chromatography here on the left. It uses large diameter stationary phase or particles, which does not require much pressure to force the mobile phase through the stationary phase. To the right of that, you have high performance liquid chromatography. Here the particles are much smaller, about 3 to 5 micron, and the pressure needed to force the liquid or mobile phase through this stationary phase is much greater due to how fine the particles are. This requires specialized pumps, which increases your cost. Finally, to the right, you have ultra-performance liquid chromatography. This often uses sub-2 micron particles, thus demanding pumps that can achieve very high pressure, around 14,000 pounds per square inch. And due to these special pumps, or the pumps designed for high pressure, you have an increased cost. Due to the much smaller particle size, you've increased your resolution that you're able to achieve in the same amount of time. By resolution, we're talking about the ability to resolve your analytes of interest. In depiction A, you see that they're a critical pair, not baseline separated. And finally, in depiction C, you can see that they're baseline separated from each other. So the ultra-performance liquid chromatograph can achieve a much higher resolution and a much slimmer or lower time. Thus, its power of doing high performance or ultra-performance liquid chromatography, because we are demanding of doing many samples in a clinical laboratory or have the demand of time upon us. So for the components of a high-performance liquid chromatograph, we have our mobile phase depicted here and with the HPLC solvent. You have the stationary phase in the middle of the picture with the HPLC column. In order to force the solvent through the column, you have the HPLC pump. If you're using a single solvent system, that's an isocratic method. Oftentimes in a clinical laboratory, we're using at least 2 solvents, thus creating a binary system. We need to introduce your sample into the flow path of the mobile phase to go through the stationary phase. So you have an injector where often a switching valve is utilized to introduce your sample into the flow path. Finally, after it elutes from the column, you need a way to detect the compounds of interest coming off. That is linked to a computational system for the data analysis, and the specimen itself is then gone to waste. Now this can become more and more complex. You can add on multiple pumps, as we discussed with binary systems, where you have multiple mobile phases, and you have multiple columns to achieve potentially the sensitivity or specificity that you need. Not to go through this, but just an example of one pump system in our laboratory, where you can see there's multiple injectors, multiple switching valves, multiple columns on different systems on this LC to achieve different methods that you need. So it can get much more complicated than the simple depiction. So what is inside these columns? What is the column chemistry? All the way to the left, you can see a normal phase column chemistry. This utilizes a polar stationary phase, a silica, for example, with a nonpolar mobile phase, such as maybe hexanes. Reverse phase is the opposite of normal phase. Now the stationary phase is no longer polar, it is nonpolar, and the mobile phase is an increasing from polar to nonpolar. Ion exchange utilizes one of the fundamental principles of chemistry. Like charges repel, opposite charges attract. So your stationary phase is charged, where the mobile phase is the opposite charge of the stationary phase. Finally, you have size exclusion. So we can imagine an example where you had a race in a busy hallway in your hospital or laboratory. The race is between you alone, and the other group is maybe 4 of your favorite laboratory mates that are all holding hands. And you ask them who could reach the end of the hall first, the one acting alone or the one linked with 4 hands. You can see that the bigger you are, you might have difficulty transversing. So in the clinical laboratory, one of the most common column chemistries utilize is reverse phase. So very briefly diving into this, what is the polar and nonpolar phases utilized for reverse phase liquid chromatography? Here we see Pauling electronegativity chart on the periodic table. Fluorine, all the way on the top right, has the highest electronegativity. It desperately wants to be like the noble gas to the right of it, neon. Juxtaposed to this, all the way to the left of fluorine, you have lithium. Lithium has a low electronegativity, meaning that it would like to give up one of its electrons to look like its partner noble gas, helium. So you can have an unequal sharing of electrons, and since electrons are negatively charged, you can create polarity. For example, here we have water, a very popular polar mobile phase. Oxygen is represented in red, and hydrogen is represented in white. So if you look at the Pauling electronegativity, you can see that there's a 1.4 difference between the oxygen and hydrogen atoms, meaning oxygen has a higher electronegativity than hydrogen, so you have an unequal sharing of electrons, or that negative charge. Because of this, you have a partial negative charge on the oxygen and partial positive charges on the hydrogen. You can now draw a pole from plus to minus, going from the bottom of the screen to the top of the screen, thus creating the polarity. Opposite of this is nonpolar compounds. So here we have a carbon-carbon bond. They both have the same electronegativity, meaning there is no difference between them. And now you are unable to draw a pole of positive or negative because there is no difference between the two. So this is a nonpolar compound. Common examples to think of this would be oil and water, for example. So how do we utilize this in our column chemistries? So depicted here, we have the inside of a column. On the bottom, we have a C18 carbon-carbon chain. This is a common column chemistry that's utilized as a nonpolar stationary phase. As the mobile phase, we have water, which is polar. Our two compounds of interest are depicted here as A and B, and if they have any hydrophobic characteristics, they'll have interactions with the C18 stationary phase. After loading, we then ramp the nonpolar solvent from mostly water to mostly nonpolar, such as acetonitrile or methanol. The hope is that you'll have a differential interaction between the mobile phase and stationary phase with A and B, so they separate at different times because they have different periods where they like to be in a certain percentage of nonpolar mobile phase. And here in the final cartoon, this is depicted as B has eluted first. It is more hydrophilic or water-loving than A because it has come off with a lower percentage of nonpolar, as depicted here as %B. B being the strong solvent in a binary solvent system, and A being the weak solvent, which is water. A is more hydrophobic than B, coming out later, requiring a higher %B. But we have achieved baseline separation between the two. So an important facet of liquid chromatography is that we have to be able to detect what is eluting from our column. These are broken down into two domains, one being non-destructive. This, in theory, means you could collect what elutes from the column again, and then thus not destroying it. A common example is UV vis, where a spectrophotometer is utilized, and if you want, the eluent can be collected again. Destructive detectors, this is unable to retrieve the specimen after it is introduced into the detector. We are highlighting mass spectrometry today. Once the analyte has entered the mass spectrometer, it is unable to be retrieved, thus it is destructive. So with that, let's talk about the other component of our hyphenated system of LC mass spec, the mass spectrometer. A mass spectrometer measures or analyzes ions by their mass-to-charge ratio. This sentence is packed with a lot of information. It's very important to the methodology. First, it measures or analyzes ions. So you have to be ionized in order to be measured by a mass spec. So you have to have a charge associated with the analyte of interest. Second, it's actually measuring their mass to charge ratio. So what is that? So below we have an example of a very simple mass to charge ratio. On the left, we have a hydrogen atom. The hydrogen atom has one proton, which has a positive charge, and one electron that has a negative charge. +1 - 1 is 0, so there's no net charge. Further, the weight of a hydrogen atom is 1 Dalton coming from the proton. The electron adds a very insignificant amount of mass. On the right, if we remove the electron, then we still have the Dalton, 1 Dalton, but we have a positive charge with no canceling negative charge. So the mass of the proton is +1 divided by the charge, which is 1. Therefore, the mass to charge ratio of this proton is 1. So let's have an example for the audience. We have phenylalanine depicted here on the left. It has a mass of 165 Daltons. If I were to introduce this into an acidic solution, I could possibly protonate the amine to become NH3, therefore carrying a positive charge and also carrying one more hydrogen atom. If I add another Dalton to the mass, I get 165 + 1, which equals 166, and the charge is 1 for the 1 positive charge on the nitrogen. So 166 divided by 1 is 166. So the mass charge ratio of phenylalanine is 166. So what is inside this mass spectrometer? A mass spectrometer consists of 3 main components, first being an ion source. As stated earlier, you have to be an ion to be analyzed by a mass spectrometer. So this area, you have the creation of ions for the method. Second, you have a mass analyzer. A mass analyzer needs to separate the analytes of interest by their mass to charge ratio, and so therefore they hit the detector differentially. And the detector is what is producing your signal at the end of the components. The detector is hooked up to a computer where the analysis is performed to get your signal, and all this is under vacuum. I often use the analogy of building a mass spectrometer to buying a car. Depending on what type of car you want to have, you would pick out the different one from the lot. For example, if you're hauling a large family, maybe you'd want a minivan. The minivan could have large doors on the side to get everybody in and out and many seats. Or if you're hauling a trailer that's very heavy, you would want a truck. It does not need to have capacity for people, but you would want to have maybe a large engine, so you'd have the towing power needed for the trailer. For a mass spectrometer, what you build is dependent on what you're intending to measure. Many of you might be familiar in the microbiology world that they use MALDI-TOF, so matrix-assisted laser desorption ionization, as the ion source, coupled with a mass analyzer of time of flight. Often in the clinical chemistry world, we are using ESI coupled to a quadrupole. So ESI is electrospray ionization. This technique has the repulsion of analytes based on similar charges, hopefully removing solvents and introducing them into the gas phase into the quadrupole. The quadrupole is the mass analyzer used, and often this is coupled with an MCP or electron multiplier stand for the detector. Further, we can add additional components. Here depicted, you have a triple quadrupole mass spectrometer, thus called because it has 3 quadrupoles, 2 of them analyzing and 1 not, as depicted as 1, 2, and 3. To work your way through the instrument, first we have the introduction of the sample coming off your liquid chromatograph. Here it enters the ESI, electrospray ionization source, where there is a differential voltage utilized in order to introduce the analytes into the gas phase and Q1. Q1 is a filtering quadrupole, so a single mass to charge can be selected, thus depicted here by a small blue dot, and we filtered out the other analytes. This blue dot is then introduced into the second quadrupole, where it does not filter, but is a collision cell. The analyte of interest is collided against an inert gas, such as nitrogen or argon, to break into fragments. The fragments are then introduced into the third quadrupole, which once again is a filtering step. Here we have depicted a blue triangle. Ultimately, this will be allowed to pass through the third quadrupole and hit the detector, producing the signal. So many things going on here. One is that you need to be a blue dot that breaks into a blue triangle, thus increasing your analyte specificity that you'd have. You can add multiple fragments. Instead of just a blue triangle, you can have a blue triangle and a Pac-Man that need to exist. And these two can be at different ratios of each other, thus 2 to 1, and that can be monitored for the potential of an analyte that's not your analyte of interest. All in all, this is called multiple reaction monitoring, where we're monitoring multiple reactions of Q1 breaking into a triangle and the blue dot, for example, breaking into the Pac-Man. It's very powerful for us in the clinical laboratory. As an example of fragmentation, we have phenylalanine again breaking into 4 different components. So for the precursor ion, the one that is filtered in Q1, you could have the mass to charge ratio of phenylalanine. And then for Q3, you could select 1 or up to 4 different compounds or fragment ions to be measured. Briefly, we'll talk about why LC mass spec, or this technique we briefly touched upon previously, is important to the clinical laboratory. Immunoassays are often utilized in the clinical laboratory, but they have several drawbacks. Here, depicted on the left, you can see we have a blue antibody connected to potentially a paramagnetic bead that a magnet could be used to help collect it. And then the green antibody potentially has some sort of signal-producing substance, an enzyme, for example, or maybe a compound that glows or fluoresces. In order to complete a immunometric or sandwich immunoassay, you need to have the compound of interest connecting these two antibodies. Here we can see we have an antibody to the compound of interest, thus shielding it from the sandwich assay, causing a falsely low result. The opposite of this is called bridging. We have a heterophile antibody that has interaction with the two capture antibodies shown for the sandwich assay, thus causing a falsely high result. And oftentimes used for small molecules, we have competitive immunoassays. We are unable to use a sandwich or immunometric assay because we have steric hindrance. The molecule is just too small to get two antibodies epitope recognitions. So here we use a competitive assay with an analyte of interest that has a tag on it, for example, fluorescence. We have the interaction or competition of these two, both the endogenous and the one in the reagents, between the antibody. So if the signal is very high, that means there is low amount of the endogenous compound. Conversely, if the signal is very low, that means there is high amounts of the endogenous compound. Some of the pitfalls for this is that since it only has one epitope recognition site, the specificity of the antibody can suffer due to just the size of the molecule, causing potentially low or unable to reach low levels of sensitivity for measurement or getting many false results. Here, conversely, we look at advantages of the LC mass spec methodology. Mainly is the increased specificity. So knowing what you're measuring is what you're measuring. We have many quality measurements within a LC mass spec methodology that all need to be positive in order to produce the signal. For example, the retention time, if you build an assay and you have it elutes between 0.8 and 1 minute, You can always measure that, and if it's outside that window, it's not your compound of interest. You have an internal standard that can be added. An internal standard is often an isotopically labeled analog of your analyte of interest, maybe replacing the carbons with carbon-13s or replacing the hydrogen with deuterium atoms. This should have very similar characteristics with LC mass spec methodology and have similar, if not the same, retention times. Also, you can monitor multiple transitions, which we mentioned earlier, with the blue dot breaking into potentially a rectangle, and a Pac-Man can be monitored. With this, you can monitor the ion ratio between those two to ensure that they are meeting what you've seen in validation. If it's always a 2-to-1 ratio and it's suddenly a 100-to-1 ratio, this gives you an alarm that it's not your analyte of interest. You have the internal standard, ion abundance. Your internal standard is added at the same amounts for all of your analytes of interest and calibrators. So seeing a doubling, tripling, or quadrupling of this can be a sign that there may be an interference with your internal standard. And finally, you have the chromatography. The chromatography can be reviewed for peak tailing, peak fronting, split peaks, multiple peaks eluting, potentially causing interference with your measurement, which can be reviewed. All of these are viewable versus a immunoassay, potentially, where you may be able to see a reaction monitor, but ultimately you're just producing a signal from a secondary interaction with the antibodies. One example of its strength is seen here with 2 immunosuppressants. The immunosuppressants, as depicted on the right, are quite similar in structure. Many antibody-based tests can have difficulty differentiating between the two. With LC only, you can see that the 2 peaks elute exactly the same. However, since they have different mass-to-charge ratios, they can be easily distinguished with an LC mass spec methodology. Next, we're going to talk about an example of an LC mass spec clinical validation. The example we're going to be using is triazole antifungal drugs. Depicted here, we have 5 triazole medications: fluconazole, itraconazole, voriconazole, posaconazole, and isobuconazole, and all have different antifungal activities to the species listed. These medications are important for invasive fungal infections, which can be deadly for immunosuppressed populations, such as transplant patients. If you in your hospital has decided that this is important because you have a large transplant population and the turnaround time that needed to adjust dosages is not being achieved by reference laboratories, you could bring this method into your lab. If you decided to do so, a review of the literature shows different methodologies available. You have the bioassays, which take almost a day to complete. They have very low analyte specificity, meaning knowing what you're measuring, and sensitivity, meaning how low you can measure, your measuring range, which is how low to high you can measure, and multiplexing, meaning can you measure more than one analyte at a time. LC alone is an hour, approximately, has better analyte specificity and sensitivity, a great measuring range, and you can measure more than one triazole at a time. Finally, at the bottom, LC mass spec by multiple reaction monitoring can have the lowest time, the best specificity and sensitivity, a great measuring range, and can multiplex. So the decision could be made to move forward with an LC mass spec methodology, which is best for your laboratory. So we need to develop the method. So here, very briefly, we have 50 microliters of the patient's serum or plasma. The proteins are precipitated using a acidic acetonitrile solution. The precipitate is centrifuged, the supernatant is taken and then diluted with our mobile phase A and then introduced into the LC mass spec. And we're using the following LC and triple quadruple mass spectrometer from Thermo. Chromatography is depicted here. B is a strong solvent, so ramping from 40% to 60% before equilibrating. These are quite hydrophobic compounds. We're using the C18 column, which you depicted earlier, about a 5 centimeter length, 2.1 diameter, and the diameter of the particles is 2.6 micron. Solvent A is acidified water. Solvent B is acidified acetonitrile, so the polar and nonpolar mobile phases. The flow rate is 0.8 mils per minute. And here's an example of the chromatography of the 5 analytes plus hydroxyitraconazole on the bottom, which is a metabolite of itraconazole. So once you have your developed method, you've done all your pre-validation work, and you feel comfortable moving forward with the establishment of this method, we can look to our regulators. CLIA it requires us to establish the following 7 characteristics: accuracy, precision, analytical sensitivity, analytical specificity, the reportable range, the reference interval, and finally, any other performance characteristic required for the test. We can think of this as kind of a catch-all. One common example I'll utilize is stability. Of course, you need to know how long from draw to analysis you can wait before you'll have degradation of the compound. So for experiment specifics, there's not much listed in the CLIA law. However, you can look at FDA guidelines for IVDs, CLSI guidelines. These are consensus-based guidelines on different areas in the laboratory. And then the medical director or director of the laboratory who's over the test. Before starting a validation experiment, we want to make sure that we have a strong pre-validation protocol. So with this, we list the details in regards to what experiments we want to perform, the ranges we want to measure, the different matrices, for example, the material we're going to use. We have the acceptability criteria of what a failing or passing experiment looks like, and then ultimately any signature or any deviation from this requires a director's signature. The validation experiments typically performed for an LC mass spec validation include ion suppression, which we'll talk about in a moment, a mixing study, interferences, the reportable range, carryover, stability, precision, method comparison, and reference interval. For this presentation, I'll just focus on one, isobuconazole, which is the analyte that comes from the prodrug listed here. It was recently brought through FDA in March of 2015, almost 5 years ago, for the treatment of these 2 invasive fungal infections. The first experiment is ion suppression. Ion suppression is the phenomenon with LC mass spec methods. In a perfect world, your analyte would be the only thing eluting from the LC column and being introduced into the ion source. Unfortunately, this is a foregone impossibility. There will be many other analytes, even if they're not direct interference, eluting at the same time from the liquid chromatograph. If they're in high enough abundance, they can compete for the ionization of your analyte of interest, or leaving the liquid phase, entering the gas phase and ion, and being introduced in the Q1. To assess if this is happening, we utilize the post-column infusion experiment. It's one of 2 common experiments used to assess this. At the very top, we have the LC pump, the injector and column, and then the ion source. Here, we're introducing patients that are free of isobuconazole after they've been extracted and ready for the method. In the bottom, we have the syringe pump, which is being teed into the post column. Here, we're constantly infusing a solution of isobuconazole. The bottom depicts the experiment. The blue line is a matrix that is a blank, such as a solvent that's being injected. In a perfect world, all of your patient samples would look exactly like your blue line that's depicted. Unfortunately, you can see that many of the black lines, all of the black lines are having a suppression around the very front of the method. This is due to many hydrophilic compounds eluting off the column, having no interaction with the C18 hydrophobic phase. If isobuconazole were to elute around the 0.2 minute mark, it would experience ion suppression. However, isobuconazole is eluting around the barred blue lines around 1.1. Here you can see that the black lines and blue lines are intertwined, and we have a depiction of 20% up and down from there, meaning it's well within. So no ion suppression is seen, or ion enhancement, which can be seen in very rare cases. Next, we have a mixing study. This is to determine a candidate matrix. We chose charcoal strip serum. This is commercially available and easy for us to obtain. We did mixing between our sample matrix, serum and plasma, and this charcoal strip serum with different levels of isobuconazole and different ratios of the mixture. The theoretical should match the experimental if the two matrices are equal. After the experiment was performed, the difference between the two was about 5%. Next, we have analytical specificity. First, we evaluated endogenous interferences common to the clinical laboratory, such as lipemia, hemolysis, and icterus. This was evaluated at 2 isobuconazole concentrations, 1 and 6, and they were mixed with patient samples or store-bought hemolysis, icterus, or lipemia to see if there's a difference between the two after they are mixed together from the theoretical to the measured concentration. At the low levels of isobuconazole, they all match within 10% or 0.1 micrograms per milliliter. At the high levels of isobuconazole, they're all less than 5% difference from each other. Next, we have interferences from exogenous sources. We looked at common therapeutic drugs. What you want to do when you evaluate analytical specificity for a medicine such as isobuconazole or any other analyte is look for isobars. Isobars are medications that may have the same mass as your analyte of interest. If it has the same mass, it may have the same mass to charge ratio. It may have a similar elution, may have similar fragment ions, and the fragment ions may have similar ratios. If all those criteria are met, you can have an interference or a false positive result with your method. Here we have the reportable range and analytical sensitivity. We purchased traceable isobuconazole that was spiked in at known concentrations, and a recovery experiment was performed. You can also perform limited blank, limited detection, and limited quantitation experiments where you have no isobuconazole in a patient sample or isobuconazole levels at the detection and quantitation where they are ran or analyzed at multiple time points to determine their variance and precision. Here we established a 0.2 to 10 microgram per milliliter range with observable error less than 5%. Carryover is another catch-all item as in number 7 with their CLIA regulations and also with some of our other regulators such as CAP. Multiple ways to perform carryover. Here's a very simple demonstration of analyzing a low concentration followed by an absurdly high concentration that you would hopefully never observe in a real patient sample, followed by the low concentration again. The means of the low concentration should match the means of the low concentration prior to the high concentration being analyzed, which is seen here with no difference. Stability is another needed item. 2 stability studies were performed. One is the unextracted, so serum or plasma that was left at room temperature, refrigerated, or frozen. The other extracted, so this is a sample that's been prepared, the proteins have been precipitated, it's been diluted with mobile phase A and ready for analysis. This is important for extractions that may be longer, that these precious samples may have to sit because there's an instrument with your LC mass spec machine. So here we established that 7 days these can be left refrigerated without the need for re-extraction of the original patient sample. That may or may not be available. In the cartoon, you have the 3 levels that we chose, and the hope is that at the end of your time frame, it will match the beginning at day 0. Precision, we performed this CLSI experiment where mid, high, and low samples are ran in this order twice a day for 5 days. There are multiple ways to perform precision, such as running your samples once a day for 20 days. The means of the 3 levels we chose are here, as well as their within run. So that's the same assay without the need of re-extraction, and then total, which is a combination of the within run and then the multiple days analysis, all having CVs less than 3%. Accuracy was evaluated with our traceable standards, but was also evaluated with a comparator laboratory, which is a CAP requirement. They perform at least 20 samples. There are 40 samples were shared with a comparator laboratory that did not have an exact method, but a similar method. We had a slope of 0.996, perfect would be 1, an intercept of 0.115, perfect would be 0, an R of 0.9927, where perfect is 1, and the difference between these 2 methods, although states away, was less than 4%. The reference intervals. So obviously a normal reference interval, as you think of with calcium, for example, you couldn't take 120 healthy individuals and run isobuconazole because healthy individuals wouldn't be on an antifungal medication. So here we can look at what are therapeutic or prophylactic levels in literature from large trial studies or from societies such as the Infectious Disease Society of America shown here, where you can establish proper ranges for your method. Thank you for listening. I just have a couple of key takeaways after our validation study. One is that the hyphenated method of LC mass spec is excellent for analyte sensitivity and specificity if you design a proper assay. Secondly, due to this excellent analyte sensitivity and specificity, this can be a very powerful tool for the clinical laboratory for a better method that doesn't that may be out there, or to a method that may not even exist. Thank you very much for listening today. And with that, I'd love to take any questions.
Thank you, Dr. McShane, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. So our first question is, besides small molecule analysis, is LC-MS useful for larger analytes?
Oh, that's a great question. LC mass spec has found a home in small molecule analysis. However, recent advancements in the last 10 to 15 years have seen this move into the larger molecule world, such as proteomics. You can see different methodologies being utilized as a cytoglobulin in reference laboratories, subtyping amyloids. Basically, these proteins are broken into peptides and then analyzed, or with the power of different mass spectrometers that are the ability to have high resolution, you can have intact analysis of proteins. So a very exciting field that's been burgeoning for a decade or two.
Great. Our next question is, what are the hurdles to LC-MS implementation?
Yeah, so if you don't already have this system, you have the upfront cost of buying one. Then, of course, it utilizes different gases that you would need, such as nitrogen or argon, so that would have to be bought or piped in. Although there is movement into wholly FDA-approved devices, many analytes are unavailable, so you need the expertise to develop and validate these assays, and also keep them running and robust.
Thank you, Dr. McShane. So we're going to wrap up with this final question. It is, what are some common issues seen with routine MS testing?
I would say one of the most common issues after a method is validated and you're running for clinical purposes is issues with sensitivity. This can be monitored with different mixtures of your analytes of interest, such as isobuconazole and a solvent system, to see if the recovery of the instrument is not behaving that day, whether it be from potential dirtiness in the fronts at the orifices, such as between the ion source and Q1, potentially a failing vacuum, so you're not removing the other atmospheric gas in the air. So I would say that was probably one of the most common issues we see is low sensitivity on the mass spectrometer.
Those are great questions. Thank you again, Dr. McShane, for your time today and your important research. We would also like to thank LabRoots and our educational sponsor, Agilent, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of your registration. This webcast can be viewed on demand through April of 2021. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.